Okay, so for part two, we're going to look at membrane transport. So don't forget to think about like what the membrane is composed of. The membrane is composed of a double layer of phospholipids. What that means is the phospholipids are going to have a great structure in a water-based environment. It's going to prevent too much water from coming in, too much water from going out. But then that becomes, like I said previously, like, it becomes a problem. How do you make sure water gets in and how do you make sure water goes out? Or anything that is water loving, which is hydrophilic, how do we get across this thick layer of fat? We can use channels. That's one of the way that we do that. But regardless, if you have channels or not, there is a natural inherent uh, property of the membrane. It's actually more of a natural chemical process that allows for the movement of atoms and molecules in a natural way. This is called passive transport. Passive transport does not necessarily require a boundary, but when we're talking about living things, it does utilize, or and maybe I shouldn't say utilize, it does happen across the membrane. There we go. So the first type of membrane transport is called passive transport. In passive transport, what you're going to do is you're going to move molecules from an area where they are higher in concentration to an area where they are lower in concentration. So we just call that from high. And if you haven't taken chemistry, anytime you put a bracket around something, it means concentration or amount. So from a high concentration to a low concentration area. The simplest type of transport is known as diffusion. And diffusion is very simple. You move molecules from a high area to a low area. This doesn't even require a boundary to do that. So let's say you're baking cookies. There's my oven, like that. You're baking cookies in your kitchen. Eventually, as those cookies bake, the smell molecules are right in your kitchen, but they will eventually go from that high concentration area to you over here studying in your room as they start to drift to the low concentration area. This is known as diffusion. You can do this for a cell. You just have to do that through the membrane, as you can see up over here in our little GIF from the Amoeba sisters. You have a high concentration on this side of the membrane, and you have a low concentration on the other side. These molecules will slowly progress through the phospholipid layers in order to move from high to low. They want to do this. It's their natural ability to do this. Think about molecules bouncing. As molecules bounce, they bounce off each other, and the more molecules in a space are going to bounce more until they get to an area where there's less molecules to bounce off of. If you're talking about hydrophilic molecules, this process will take more time because as soon as you hit the middle, you might end up getting shot back out. But if you give it enough time, you might make it all the way through, even if you're hydrophilic. So some water does go through the membrane back and forth because eventually some molecules will, as you have this high and low concentration area. However, you can use something called facilitated diffusion instead. In facilitated diffusion, you have to have a boundary because you have to facilitate the diffusion. And to facilitate that through the boundary, what you're going to do is you're going to use, oh, I'm sorry, I'm trying to draw this too fast. You're going to use a protein channel, like in our picture right there, like in our other animated GIF or GIF, however you want to pronounce that. Just sort of depends, right? So if you have that channel that goes through, let me erase that part, it facilitates the movement of substances that wouldn't otherwise move through the membrane easily. It's just facilitated diffusion. You will see that there are some cells in the human body that have those protein channels that allow for things to go back and forth to facilitate that movement. Oftentimes it's water facilitation, but it doesn't have to be. The third one, which is probably the most complicated of all passive transports, is called osmosis. It's oftentimes what students try to use in order to study, but it doesn't make any sense because osmosis is the movement of water. It is only water, okay? You have to have a membrane because you can't move water from one place to another place if both places are water. I can't move water from one area to another area if both places are water. I have to have something between them and I have to have a difference in water concentration. So I have to have that membrane for osmosis. The other thing is you also have to still have a gradient. So you have a gradient of water, but what the other thing is in the water is the solute. So really you have to have two solutions. So you have to have one solution on one side of the membrane and another solution on the other side of the membrane. Yeah, okay. So you have two solutions across a membrane. And this is still diffusion. So you have to move water from a high concentration to a low concentration through a membrane. What gives you the concentration differences is how much of the solute you have in water. And that's what this other GIF is showing you here. So you have like a hypotonic solution, an isotonic solution, or a hypertonic solution. These are the solutions that are surrounding the cell, in this case, our red blood cell, and it can change the shape or tonicity of our red blood cell. That is why it's called a hypertonic or hypotonic or isotonic solution. In a hypotonic solution, you're going to have here high H2O. Yes, high H2O concentration and a low solute. The hypo actually goes with the solute. So there's under solute. In an isotonic solution, iso means same. So you have the same amount of water on the outside and the same amount of water on the inside. You see why our cell has movement across the membrane, but it's not in a great amount. So our cell is not changing in shape. In our hypotonic solution, because you have more water on the outside, diffusion tells us higher concentration goes towards lower concentration. So I'm going to start to move more of that water towards the inside of the cell. And as you move more and more water towards the inside of the cell is going to get bigger and explode. In a hypertonic solution, you have low water on the outside and high solute concentration. So that means you have way more salt on the outside, or any other solution is higher on the outside, and you have lower amounts of water on the inside of the cell because you're going to go from, sorry, what did I say? You're going to have lower amounts of water on the outside and higher amounts of water on the inside. So you're going to go from your high concentration to your low concentration and pull that water out of the cell. So this is a very important concept for many of you to understand because this is something we sort of talked about in class a little bit, maybe not in great detail, but one of the things you do in your healthcare career is you're probably going to give your patient an intravenous solution. Okay, you're going to give them an IV. You give them an IV so that they get fluids, water, they get salts, they get everything that they need in order to make sure they maintain the health of their body, possibly when they're not drinking regular amounts of water. Think about it. If you go to the hospital, your patients aren't always constantly drinking water. How do I make sure they're hydrated? How do I make sure that they're not sweating out all their salts? I have to give them an intravenous solution. In this case, you want to understand where is that intravenous solution going and how is that intravenous solution given so that you don't change your patient's cells, not just their red blood cells, but also their tissue cells. So here's my little map of the different areas that would be inside of the human body when we look at going from cells to like a tissue layer. You can see, oh, we have our intracellular compartment and we have our extracellular compartment. No different than what I wrote right here. I have my extracellular part and I have my intracellular part. In this case, intracellular is actually the space around the nucleus, not the nucleus itself. That is the cytoplasm. That is where our cytosol is. Our cytosol is going to be a solution. This is called intracellular fluid or cytosol. It's water plus salts plus sugars plus a many combination, like a combination of things that helps us maintain the health of our cell. But now you can see that we have some cells that are kind of close together, some cells that are far apart. So we go back to our generic eukaryotic cell here, we can do that. Some of your cells are going to be almost touching, pretty much they will be touching. And what that means is there's not a lot of space between those cells, but there has to be enough space for there to be a little bit of some extracellular fluid. It's just not going to be a lot. Some of our tissues have cells that are widely spaced apart, and they have more extracellular fluid going between them. So regardless, you are always going to have a little bit of fluid around, okay? Depending on what type of tissue you have, you will either have an interstitial fluid. That interstitial fluid you see is right here. It goes between the two cells. Usually this is for cells that are kind of closer together, and there's not a lot of fluid bathing them. Another option is you can have something called plasma, which all of you know. Plasma is the liquid component of blood. This is just to show you that you always are going to have some fluid wrapping around your cells. So why is this important? Well, when I put an IV right there, there's like kind of my needle, into the blood supply, what is going to happen is that you're actually adding fluid plus solutes right here. So let's just say we're going to hook up our IV right there. Okay. I don't need it to be a 5. Sorry. If our IV hooks up right there, and we just put, let's say, a hypotonic solution. A hypotonic solution we see here, I wrote, is low in solutes. Let's just imagine this is pure water. Is our hypotonic solution. So you have somebody who's dehydrated, you hook them up to an IV, you give them pure water. Now, that means you have pure water around your red blood cells. But inside of your red blood cells, it's usually a 0.9% saline solution, so 0.9% salt. In that case, you have more water on the outside and less water on the inside. Your cells will start to expand. That will also lead to more water leaving your blood supply and going to the interstitial space, where now you have more water on the outside and less water on the inside. This is going to cause all of your cells to start swelling. That is not something you ever want to have happen. This is why pure water is never given, really, in these situations, unless you are severely dehydrated and your cells are like really shriveled up, then they might give you pure water, but it's not the best option. Instead, they give you an isotonic solution because if you have 0.9% solution on the outside and 0.9% of solution on the inside, water will freely exchange across the membrane. You can start to hydrate with the same amount of stuff. I shouldn't say stuff, the same amount of solution that you would normally have in blood, and it will hydrate you without causing cell changes. This is also why, like on a really hot day, if you've sweated out a good majority of your liquid and a good majority of your salts, if you drink pure water, you're now putting mostly just water back into your bloodstream and you don't have a lot of salts. It will also cause swelling. You see that on like really hot summer days where your hands and your feet might swell. That is because you're not getting enough salts to help you replenish the salts that you've lost through sweat. All right, the last one here we see where you have a solution that is hypertonic, right? So that's our situation over here. So imagine if our IV, you give your patient something that's not 0.9% saline, you give them 1% saline instead. So here you can think about 0% sodium chloride. That's what saline is. Here you can think about 0.9%. And here you can think about like 1%. I know that doesn't seem like a lot. We didn't go up a lot, but it doesn't take a lot. So now you have more salt on the outside. If it helps you, you can also think about this as, water always travels towards high salt concentration. Think about it. Sometimes I like to think about it like if you eat really salty potato chips, you want to drink more and more water because now that salt has accumulated in your bloodstream and you have less water, so you want to balance it out. That's the case here too. Inside of your red blood cells, 0.9% saline. So since there's 0.1% saline on the outside, there's less water on the outside. So you start to pull that water outside of your cells. This is why it's given to a patient that's swollen, because a patient who's swollen is like this red blood cell, and you want to draw that water out by putting it in a more salty environment. This is also given with care because you don't want to remove too much of that fluid and essentially dehydrate your patient. Okay. This is the reason why you wanted to know all these different areas of like inside and outside the cell and thinking about how cells can change as a result of that. And here I was going to draw like all these hypotonic, isotonic solutions, but I think I did a pretty, reasonable job, hopefully explaining that. And hopefully my picture is not above all of these animated uh, GIFs again. So the last one is we're going to look at active transport. So passive transport is important because every cell does passive transport, but active transport is also important because active transport is going to help us in several situations that we're going to see this semester, in particular when we look at neuron physiology and muscle physiology. Passive transport's always working, and then we need to use active transport to counteract a couple of things. So there's only one type of active transport. It's called active transport. In this case, you need a membrane. You need a membrane protein. I mean, you can't have a membrane protein without a membrane, so that's why you have both those things. Now, instead of you moving from a high area to a low area, you're going to pump against that. So in this case, you're going to pump from a low concentration. Oh, maybe I should put from. To a high concentration area. So you're actually going in a backwards direction, in an unnatural direction. And when you do this, it requires ATP or energy. This is why I say pump. Because passive transport doesn't require, you just let it happen. It just will happen on its own. But when you need to do this actively, you have to input energy into it because those molecules do not want to be in that high concentration environment, which is why they're passively moving to the low concentration environment. And you have to put energy in to push them to an area they do not want to be. So we got two examples down here. So here you see this is the high concentration, and we have the low concentration area, and you see how my energy is pushing these molecules from the low concentration area back to the high concentration area. And diffusion will tell these things to go right back in place again, and they'll start to move across the membrane again. And then the pump will come in and pump them right back out. Same thing on this other side. Here we have a low concentration area and we have a high concentration area like that. So we just pump them against the gradient. That's what it's called. Okay. When you go from high to low, it's a gradient. When you go from low to high, it's against that gradient. I like to think about active transport. I used this example in one class as like this. You're in a boat. There's my boat. Okay, it's not the greatest boat, but hey, it floats. There's my boat in water. Oh no, I sprung a leak in my boat. That is diffusion, where now the water from the high concentration area goes to the low concentration area. Sorry, that says low, but it doesn't look like low. You're right there. It goes from to the low concentration area. That is what diffusion tells us. You don't need any energy to that. All these molecules want to get away from each other, so they'll go to the low concentration area. But you're in this boat. This is you. Now you have to make sure you don't sink. So the only way that you make sure that you don't sink is you're going to get yourself a nice bucket and you're going to try and push that water back out of your boat from the low concentration area to the high concentration area. This is going to require you to constantly be throwing stuff out of that boat, and it's going to require a lot of energy so you don't sink. That's why it's active, because you actively have to do something. You will see that later on in the semester in what's called the sodium-potassium pump here, where sodium is going to be pushed to one side, to the outside, the extracellular, because of energy. And then we're going to see potassium from the outside comes in and is pushed to the inside or the intracellular because of that same energy molecule. That's going to create a gradient where we have high sodium on the outside and high potassium on the inside, something that we desperately are going to need for our neurons and for our muscles to work properly. So this is why you guys have to understand transport, because later on, I can't go over this again. It's a good idea for you to know this at the beginning, and then as we progress through class, we will use that again. All right, last part is going to be, we're going to look at some structure function stuff in another part of this video.